DNA, or deoxyribonucleic acid, contains all of the hereditary or genetic material in humans and most other organisms. So the DNA is what lets people know what genetic material is in a person or is in a certain creature or other organism. Now each cell in an organism has the same DNA. So if someone were to get a piece of your hair and a fingernail and a piece of skin and look at it very closely and look at the DNA in all three would be the same, and it would let whoever was looking at this know that all three of those articles came from the same person. This is the same for other organisms. Each cell in the organism's body is going to have the same DNA pattern so that you'll be able to tell that it came from the same creature. Most DNA is found in the nucleus of a cell. There is some DNA in the mitochondria, and then it is known as mitochondrial DNA, or it would look like this if you saw an abbreviation for it. So if you see the little MT before DNA in the abbreviation, that's because it's mitochondrial DNA and it's found in the mitochondria instead of the nucleus. But most DNA is going to be in the nucleus. Now, that important hereditary information found in DNA is stored as a code made up of 4 chemical bases. We've got adenine, guanine, cytosine, and thymine. And each one of those bases is only going to pair with one other base. They can't all pair up with any of the other 3. So adenine pairs with thymine, and guanine pairs with cytosine to form base pairs. And a way that I remember that is that the ones with the straight lines in their abbreviation letter, the A and T that can be made with straight lines, go together, and the ones that require a curved line go together. And that helps me remember that adenine pairs with thymine, these straight lines to form A and straight lines to form T, and guanine goes with cytosine because of the curved shape that it takes to make each of those abbreviation letters. The sequence of the adenine, thymine, guanine, cytosine pairs determines how the organism builds and maintains itself. So it's important to think about this. Not every organism or every person is going to have the exact same sequence of base pairs. And you might think of the alphabet. We use 26 letters to form all of our words and sentences, but the way that they're arranged, the sequence of those letters, is what lets us know what word is being communicated, what sentence is trying to be communicated. And each word has a different meaning, even though it's made up of the same 26 letters or some subset of those letters. So it works the same way. You're going to have some sequence of adenine, thymine, guanine, cytosine, and those base pair sequence is going to let you know, or let a scientist know, what that organism does to maintain itself, what kind it is. Is it functioning properly? Is everything in the right order? What does that order tell us? The same way that seeing CAT tells us that that is the word cat, and it's talking about a mammal with pointy ears and whiskers and a tail that's kept as a pet. Let's look at the next part. Each base pair, so our adenine-thymine pair or guanine-cytosine pair, is going to attach to a sugar molecule and to a phosphate molecule. And once those attach, once a base pair attaches to the sugar molecule and phosphate molecule, it forms a nucleotide. And these nucleotides form two long strands that spiral into a double helix. That is the shape that DNA takes on. And I did my best to give you all an example of a double helix. It kind of spirals around. If you were to curl a ribbon around my pen, it would form that kind of double helix shape we're talking about. And you should think of a twisted ladder with the base pairs as rungs. So if we were looking at this picture here, this would be our double helix shape that's spiraling around, and each one of these little rungs on the ladder would be a base pair. So we would have adenine and thymine together, and they would be paired up. And then here you would have guanine and cytosine paired up. And then you would have adenine and thymine, guanine and cytosine, all the way down the ladder, where you've got your sugar molecules and phosphate molecules that bound to your base pairs, making up the sides of the ladder here. And you just have this long DNA chain, and then the sequence that you see is going to let you know how that organism builds and maintains itself, because not every creature's DNA is going to be the same. Everyone has a very unique DNA pattern. DNA can replicate or make copies of itself by splitting the ladder in half. So if we're looking at this and we just decided to go through and cut this ladder in half, we would be separating those base pairs all the way down. So then you have AG, and any other base pairs you've got. So we know we've got adenine, guanine, and because we know what's on one side of the ladder, each strand or half of that ladder serves as a pattern for duplicating bases. Since we know we've got adenine here, we know the thing that has to pair with it is thymine. If we've got guanine, cytosine has to pair with it. Since in DNA, adenine and thymine always pair together and guanine and cytosine pair together, you will know, or the cell will know, okay, I've got this DNA, I've only got half of it, but it's a pattern. If I have adenine, I need to add thymine to the other side. If I've got cytosine, I need to add guanine to the other side. So the cell will know, based on that half, that strand that it gets from DNA, how to make a complete DNA molecule and be able to form that double helix and complete the DNA strand. And each new cell needs an exact copy of DNA from the old cell. Because remember, each cell in an organism has the exact same DNA. So if the DNA is replicating itself, it needs to be giving an exact copy to a new cell so that this can continue, and the organism is going to continue to have the same DNA in it. So you will see a lot about DNA in biology, and it's important to remember that DNA is the hereditary or genetic material. It's usually found in the nucleus. And remember which bases pair with which. Your straight lines in adenine, A, pair with your straight lines in thymine, T, and your curve G for guanine goes with your curve C for cytosine. DNA is the hereditary or genetic material that is usually found in the nucleus, sometimes in the mitochondria. The most important thing to remember is that it's where all the genetic material is going to be found. It's a big identifier for a cell or for an organism. Kingdom Animalia. Animals have several characteristics that set them apart from other living things. Animals are eukaryotic and multicellular. So where other kingdoms can have creatures that are comprised of just one cell, animals are going to have more than one cell. They are also heterotrophic. This means they cannot create their own food and must rely on other sources of nourishment. So they're going to eat other plants or animals. They're not going to be able to create their own food with sunlight like plants can. Animals do not have cell walls, but they may have an exoskeleton, which is like a hard skeleton they secrete onto the outside of their body, or a shell. Now, they don't have to have that. Obviously, we don't have a hard exoskeleton or a shell, but Some animals like your crawfish or lobsters would have an exoskeleton, or things like snails have a shell. All animals are motile. This means they can move, even if only during certain stages of life. So some animals may only move a little bit or very slowly, but all animals are motile, even if it's just for a short period of time or during one stage of their life. Nearly all animals undergo some form of sexual reproduction, but some animals are also capable of asexual reproduction. And this is possibly, and a lot of times, through parthenogenesis, which is when a fertilized egg is produced without any mating, budding, or fragmentation. So it is possible, but most of the time the animals are going to undergo sexual reproduction to produce offspring. Animalia is also known as metazoa, as far as a kingdom name goes, and it is divided into the subkingdoms, Parazoa, which consist of sponges and trichoplax. Sponges, I'm sure you can think of, but trichoplax kind of looks like a puddle you spilled on the floor. It's very flat, but it is still an animal. It does have differentiated cells. It even has some eye cells on it. So Parazoa, such as the sponges and trichoplax, have differentiated cells but no distinct tissue or tissues or body symmetry. So they're kind of oddly shaped. Where most animals you would be able to see some symmetry, there is no symmetry as far as Parazoa are concerned. Now, all other animals are put into the subkingdom Eumetazoa. And these have 2 or 3 distinct layers of cells with differentiated tissues and either radial or bilateral symmetry. And what that means is that you can divide them a certain way. If they have radial symmetry, you would be able to divide them any way going in a circle, and the halves would match up. So a jellyfish would be an example of that. If you divided the jellyfish here, if you could divide it across here, if you could divide it across here, going any way in a circle around the top of the jellyfish, down across the top and down through all the tentacles, if you cut it, it would be more of a circular shape, and anywhere you cut it, it would be the same on either side. So it's symmetrical. And it would have radial symmetry because there isn't just one symmetrical line you can cut along. There's not just one line of symmetry, there are several. The same goes with a starfish. You could cut it along a line going this way, 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 or a line going this way. If you go all along that circle, it can have symmetry. It can have all these different lines of symmetry, and what's on the other side of the line of symmetry is going to match what's on the first side of the line of symmetry. Bilateral symmetry, you can only divide the body in half one way. So people have bilateral symmetry. You can divide straight down from the top of your head to between your 2 feet, and the 2 halves should match up pretty well, at least as far as body type and tissues are concerned. So people have bilateral symmetry. You can divide straight down the middle and the halves should match up pretty well. Butterflies or moths are another example that should match up. Cats, dogs, a lot of animals display bilateral body symmetry, but there are some that have radial symmetry. But these are all Eumetazoa. Remember, the Parazoa is not going to have any kind of body symmetry because it doesn't have distinct tissues, and it is not going to have a distinct body shape every time. It's going to be different. So whenever you are thinking about kingdom Animalia, remember these certain characteristics that set them apart. They're eukaryotic and multicellular, made of more than one cell. They are heterotrophic. They cannot make their own food and must receive nourishment from other sources. They do not have cell walls, but may have an exoskeleton or a shell, and all animals are motile. They are able to move, at least at some stage in their life. Kingdom fungi. Fungi are a group of eukaryotic organisms. They used to be classified as plants, but genetic studies show that fungi are actually more similar to animals. Now, one reason that fungi were originally classified with plants is because they have a cell wall, just like plants do. But fungi have cell walls made of chitin, while plant cell walls are made of cellulose. So one of the differences that set fungi apart from plants is that their cell wall was composed of something completely different. Fungi can reproduce asexually via vegetative spores, or they can reproduce sexually via meiosis. A fruit body, such as a mushroom or mold, is formed where spores are created and released to begin new colonies. So when you think about a plant and how it produces a fruit, and that's where the seeds are that carries on life, it's kind of the same thing, except fungi and plants are not the same kingdom. So a fungi's fruit body is going to be in the form of a mushroom or mold, and instead of having seeds, it's going to produce spores that are released, and the spores go out and they create new colonies of fungi. Fungi include yeasts, molds, rusts, and familiar mushrooms. Something to remember is that slime molds and water molds, even though they are molds, are not grouped with true fungi. They used to be, but they are no longer considered true fungi because they just don't fit into this kingdom. So even if you see the word mold, that doesn't mean that it's definitely something that fits into kingdom fungi. Fungi grow in soil, on dead matter, and is symbionts of plants, animals, or other fungi. And being a symbiont means that a fungus can live on a plant, on an animal, or on some other fungus. So it can live on it. It could be a neutral relationship where that fungus lives there and it doesn't affect the plant, animal, or fungus that it's living on. It could be something positive where they're both getting something out of it, or it could be something negative where the fungus growing on that plant, animal, or other fungus is causing a problem. For instance, on certain rice plants, if a fungus grows on it, it can cause a disease that causes all those crops to go bad and not be good for consumption anymore. So it can be a bad relationship, or it can just be something neutral, or it can be something that's helpful for both the plant, animal, or fungus and the fungus that is growing on one of those things. And then a fungus doesn't always have to be a symbiont. It could just grow in soil or on dead matter all by itself. Fungi uses can include an essential role in decomposition. So when fungi grow on dead matter, they're going to help decompose that dead matter. It can be a direct source of food. Familiar mushrooms would be direct sources of food that you could eat, though not every mushroom, because fungi can be toxic. So don't think that every mushroom you see is all right or every fungus is going to be helpful. Fungi can be used as a leavening agent for bread. That's a yeast. Fermentation, such as in wine, beer, and soy sauce, and that would be yeast again. Antibiotics, enzymes and detergents, biological pesticides that could kill certain weeds, and then remember, fungi has a lot of uses, but it can be toxic. So kingdom fungi is going to consist of eukaryotic organisms, and they're going to have cell walls made of chitin. They can reproduce asexually or sexually, and they are going to include yeast, molds, rust, and familiar mushrooms. Kingdom Plantae. Kingdom Plantae includes green algae, chlorophyta and charophyta, Non-vascular plants, such as liverworts, hornworts, and mosses, and non-vascular means they don't have any kind of veins to carry nutrients throughout the plant. Vascular plants, or tracheophytes, which means these do have veins that carry nutrients and water throughout the plant. And vascular plants include club mosses, ferns, horsetails, and seed plants, which are also known as spermophytes. And the seed plants include gymnosperms, such as the conifers, cycads, and the ginkgo tree, and these carry their seeds on the outside. The conifers would be like your spruce and pine trees, which have a cone, and that cone holds the seeds, and you can actually see the seeds coming out of it during the time of year where these seeds are coming out and the trees or plants are trying to reproduce. Angiosperms, or flowering plants, have their seeds on the inside in an ovary of the flower, and so both of those are seed plants. They just carry their seeds differently. The cell walls of plants consist primarily of cellulose and lignin. And lignin gives structure and strength to a plant cell wall, especially in trees, and it plays a large role in conducting water through the plant. Most plants engage in photosynthesis, which is where plants take in energy from the sun and use it to carry out its own processes. But some plants have lost that ability and must obtain their nourishment heterotrophically, so they can't make their own food. Autotrophs, such as those that carry out photosynthesis, are plants that can carry out photosynthesis. They can create their own food. But heterotrophs cannot create their own food, and they must get their nourishment from other places. So there are some plants that cannot carry out photosynthesis and must get their nourishment, their food sources, from other places. They can't make their own food. Green plants produce most of the world's molecular oxygen, and that would be done through photosynthesis. Plants can also serve as foods, such as grains, fruits, and vegetables, as ornaments rather. People have plants around their houses or in their offices that are just there for decoration. They're there as an ornament. They're not there for any specific purpose. And they've also been used as medicines or drugs. And there are some plants that can be used in a harmful way and a lot that can be used in a helpful way. And while you probably would go to the doctor if you needed a medicine, for a long time, people didn't have doctors, and they used people that knew about herbs and plants, and those people would take care of them. And even once we had doctors would rely on a lot of plants, and a lot of your medicines that you would take over the counter now are created from plants or in a lab based on what the plant cellular makeup was. So plants play a large role in your medicines even today, even if you aren't getting them straight from some plant that is growing out of the ground. And the scientific study of plants is known as botany. So if you are interested in studying plants, you could become a botanist. There are lots of different categories in Kingdom Plantae, and the differences are based on if they are a green algae, if they have veins that help transport nutrients, and then you have different ones within those. So the vascular plants are the ones you're going to see the most of, and you will probably think most often of those that look like your flowering plants or your trees. Keep in mind that the cell walls of plants are made up of cellulose and lignin, where in fungi we had them made up of chitin. So that is a key difference between a plant and a fungus, is that the cell walls of plants are made of cellulose. Most plants are going to engage in photosynthesis, and plants have lots of uses, both as food or as an ornamental or decoration purpose or for medicines. Meiosis. Sexual reproduction in eukaryotes involves a form of cell division known as meiosis. It has several different stages that are carried out to let one diploid germ cell result in 4 haploid gametes. And diploid means that it's going to have multiple copies of the genetic information, where haploid means it's going to have 1 copy. So, in this first part, this would be interphase, and it's kind of before meiosis starts, but it has to do with it because this diploid germ cell contains genetic information from both a maternal and paternal parents. So you've got female and male genetic material in this one diploid germ cell, and it's getting ready to divide to carry out sexual reproduction. So we have replication going on in here because each chromosome is going to double itself so that there's extra genetic material to carry out this process. In the next phase, we have prophase, and the replicated genetic material is going to line up in homologous pairs, which means that each one is the same genetic material. It's an exact copy, and they're going to cross over a little bit at these points here, and where there's crossover, they kind of swap a little bit of genetic material. So each one of these pairs is going to take a little bit of the other pair, and that is called crossing over or recombination. So you're just kind of jumbling up the maternal and paternal bits before the division starts to take place. Then we move on to metaphase. And in metaphase, your centrosomes are starting to pull the homologous pairs apart. And then we move on to anaphase. And you can see they actually are getting pulled further apart. They're not crossing over anymore, and they're starting to move to opposite sides of the cell. And these are all prophase 1, metaphase 1, anaphase 1, because this is kind of a 2-step process. Then we keep going, and we've got telophase 1. In telophase 1, there's the cleavage furrow that's preparing for the cells to separate and become 2 distinct separate cells. There is a nucleus being reformed. The microtubules that have been connecting the chromosomes and pulling them apart have receded, and the genetic material is encased in a nucleus again. And then this last part is cytokinesis 1, where the cytoplasm is split, and there is a definite split made between those 2 cells. So now there are 2 diploid daughter cells. So we had our diploid germ cell. The DNA, the genetic material, was all replicated, so there was twice as much. We went through this cell division, so now each one of these daughter cells is a diploid daughter cell, and it has twice as much genetic material as it needs to be a haploid gamete. So we're going to go through what is known as meiosis 2, the second step in this process. So we have prophase 2. And in this phase, you'll see we've got the 2 daughter cells, and now they're kind of repeating the same process that happened up top. So there's no replication because that was an interphase, but they are going to start having the centrosomes move down to either side and start moving out to opposite poles of the cell and start pulling the chromosomes that way. In the next phase, we've got metaphase 2. You can see that the chromosomes have lined up along the equatorial plate in both of these daughter cells. The microtubules are connecting them to the centrosomes, and they're going to start pulling apart. So in our next phase, anaphase 2, you can see that actually happen. The chromosome pairs have gotten pulled apart, and now these individual chromatids are on separate sides of the cell, and they're still going to be kind of being reeled in by the microtubules at this point. And we move on to telophase 2, where we've got a nucleus forming around our new pairs of genetic material that have both the mother and father genetic material in each nucleus. They're all split up now. And we have the cleavage furrow again. The cells are getting ready to divide permanently. Your centrosomes have separated to the opposite sides still, and the microtubules have retracted. And then we go to cytokinesis 2, which is where the cytoplasm divided between these two new cells and these two new cells, and now we've got 4 haploid gametes, which means that they each just have one set of genetic information, and they're going to need a partner to be able to reproduce. So these gametes are going to be ready to be fertilized, and when they are fertilized, a diploid zygote will be formed. So these gametes are ready to be fertilized, and if they are, then they can go on to create a new eukaryotic organism. So meiosis does have a 2-part process, and it goes through the same phases as mitosis, but they're a little bit different because instead of just getting an exact copy, you're mixing genetic material from 2 different organisms, a maternal and paternal parent, and at the end you're going to have the 4 haploid gametes that each has genetic information from both parents. Mitochondria. When you see the word mitochondria, you're looking at the plural form of the word. The singular form is mitochondrion. So whenever you see those, don't be confused or think you're looking at a different word. It's just the singular and plural forms. First, let's talk about the structure of mitochondria, and then we'll talk about its functions. So the structure of a mitochondria. If you see it floating around in the cytoplasm, it kind of looks like a jelly bean. If you can see a cross cutout of it, then you're going to see this little squiggly membrane going through the middle there. So let's look at what each part of the mitochondria is. This outer layer that you can see is called the outer membrane. Next, this space in here between the outer membrane and this next line is called the intermembrane space. And then this next line is the inner membrane. So that makes sense, right? You've got your outer membrane, your inner membrane, and then the space between it is just the intermembrane space. Outer, inner membrane, intermembrane space, the space between the two. Next, you've got the space created through all these little zigzaggy patterns of the inner membrane, and the spaces that are created here and in there and down in here between where it zigs and zags, and you've got that space between each little fold. That is called the cristae. And then, last but not least, everything inside that inner membrane is called the matrix. So there you've got your structure of a mitochondrion, the basic parts of it, and it's important to know that they are found in nearly all eukaryotic cells. So that's not all, but most eukaryotic cells, and that's going to be your larger cells, the ones you find in plants and animals. And within the cell, it's going to be found in the cytoplasm. So you will see mitochondria floating around in the cytoplasm. And you could have one or you could have lots of mitochondria. It kind of depends on how big the cell is. But the basic structure is going to be outer membrane, intermembrane space, inner membrane surrounding the matrix with the cristae forming in the folds between that inner membrane. Okay, let's look at the functions. Mitochondria produce adenosine triphosphate, which is known as ATP. So if you see ATP, it's an energy source and it's called adenosine triphosphate. But a lot of times this is not going to get all typed out. You're just going to see ATP because it's a very common abbreviation in biology. Now, ATP is produced through cellular respiration, which is also known as aerobic respiration. And that's because it involves oxygen. It's basically the cell's breathing cycle where it gets to take in oxygen and use it. So aerobic respiration or cellular respiration are basically the same thing. It just whoever's typing or writing the paper or information you're looking at may use cellular or aerobic, but basically it's going to refer to the cell taking in oxygen. Now, the set of reactions that produces ATP is known as the Krebs cycle. So, the mitochondria is involved in the Krebs cycle because it goes through some of those reactions to produce ATP. And the mitochondria is sometimes called cellular power plants or powerhouses because they create the cell's primary energy source, which is ATP. So since they create the primary energy source that the cell has to use, they are called cellular power plants. They also can regulate the cell's metabolism, and they are involved in cell division, cell growth, and cell death. So all of these processes require energy, and since the mitochondria is producing the primary source of energy, it makes sense that the mitochondria would be involved in a lot of these processes. It has a lot of important jobs, but the most important is going to be producing ATP because that is the primary source of energy for the cell, and the mitochondria is what produces it. So it's very important to remember production of ATP, power plant of the cell, for mitochondria because that is what you're mainly going to see it associated with. Now it can also do these other things, regulating metabolism, being involved in cell division, growth and death, but main thing to remember is producing ATP. And you've got your structure, so remember outer membrane, intermembrane space, the space between the two membranes, inner membrane, cristae between those folds, and the matrix. Remember that mitochondria are going to be found in eukaryotic cells, and they're found in almost all eukaryotic cells, but not every single one. And most eukaryotic cells will have at least one, but they could have a lot of mitochondria, and you're going to find them in the cytoplasm. So just remember, if you're looking at a cell diagram, you're looking for that little jelly bean shape, and if you're thinking about what does a mitochondria do, think about producing energy, producing ATP. The mitochondria is your power plant of the cell. Mitosis. Eukaryotic cells reproduce asexually by dividing into two genetically identical daughter cells. Now they do this by the process of mitosis. So let's look. This first phase is called interphase, and it's not really considered a part of mitosis, but it's usually discussed with mitosis because it happens at the beginning right before mitosis happens. It's the build-up to mitosis. It's a period of cell growth and DNA replication. So the cell gets a little bigger, the nucleus gets a little bigger, the DNA replicates, so there's more of it for the mitosis that's about to happen. Chromosomes are in the form of uncondensed chromatin. And the nucleolus may still be present. So this would be our uncondensed chromatin. This darker dot here would be your nucleolus that you could still see. And these little pieces here would be your centrosomes. So whenever you see them along the rest of mitosis, remember these are the centrosomes. Okay, first stage of mitosis is going to be prophase. In prophase, the chromatin condenses into discrete chromosomes. So remember, it had gotten, we had uncondensed chromatin that the DNA had been turned into. In prophase, it condenses into discrete chromosomes. So instead of a bunch of DNA squiggled up, you've got discrete chromosomes that kind of look like little squiggly X's. The nuclear membrane breaks down. So where we had a complete nuclear membrane here, it starts to break down a little bit. And sister chromatids are joined by centromeres. So what they mean by that is this would be your chromosome, and these sister chromosomes or chromatids are joined by these centromeres. So this would be your centromere. And then each of these would be sister chromatids that form the chromosome. The centrosomes are going to move to opposite poles of the cell. So remember, we just kind of had them in the cell here. Now they've moved to opposite sides of the cell, so they're opposite each other and they're kind of equally spaced in the cell. The chromosomes have paired up to form sister chromatids connected by a centromere. And now we're ready to move on to our next phase, which is metaphase. In metaphase, the chromosomes align at the equatorial plate. So where they're kind of just all over the place, they've lined up at the equatorial plate, which means that it's equally distanced from both of those centrosomes that have spaced themselves out at either pole. The microtubules attach sister chromosomes to the centrosomes. So you've got your centrosomes, and these are your microtubules that are attaching the chromosomes to the centrosomes. They've lined them all up along this equatorial plate. And now we're ready to move on to the next stage of mitosis, anaphase. In anaphase, the centromeres divide. So remember, your centromere was that little center here that was kind of holding the two chromatids together. So in anaphase, they divide. The microtubules shorten, so where they're longer, they shorten up, moving paired chromosomes to opposite sides of the cell. So we had these paired chromosomes, and now the microtubules are shortening. They're moving those individual chromosomes over closer to the centrosomes now. And then you're ready for the last phase of mitosis, which is telophase. In telophase, the cytoplasm divides along a cleavage furrow. And so you can see how it starts to kind of dip in. It cleaves in half, and so there's that cleavage furrow, and that's going to show you where your cytoplasm is going to eventually split completely off and form two new cells. The chromosomes condense to chromatin. Remember, that's what we had up here. We had all the squigglies. They're going to just start condensing to chromatin, not a discrete chromosome anymore. and the nucleolus reappears, and the nuclear membrane is going to be reforming around a clear nucleus. So you see the nuclear membrane is forming around where your condensing or your uncondensing chromosomes are, and your nucleolus has reappeared. So once these cells split off, then this is going to be a nucleus again that looks much like this, with the uncondensed chromatin and the nucleolus in the center. Two daughter cells, as we discussed at the beginning, two identical daughter cells will be formed once cytokinesis is complete. And cytokinesis is that process of the cell being cleaved in half to form two new identical daughter cells. So this is mitosis. It is how a eukaryotic cell can reproduce asexually. It just divides into two identical daughter cells. It goes through interphase, prophase, metaphase, anaphase, and telophase. So it starts off with this uncondensed DNA, this uncondensed chromatin genetic material. The chromosomes form little pairs, and the centrosomes come to either side. The centrosomes attach to the centromeres with microtubules, and those chromosomes line up along the equatorial plate. Then they split apart, starting to look like two new cells now, and each part of the chromosome pair goes toward a centrosome so that they are split equally across the cell. And then they split apart, forming a new nucleus with a nucleolus, and cytokinesis is going to be complete once this cell finishes cleaving in half. So just remember that eukaryotic cells can reproduce asexually by mitosis. Nucleic acids. Nucleic acids consist of a linear chain of nucleotides. So that leads us to ask, what is a nucleotide? Well, a nucleotide is going to consist of a nitrogenous base, which could be a purine, such as adenine, which is usually abbreviated with A, and guanine, which is usually abbreviated with G. Another nitrogenous base is a pyrimidine. Another type of nitrogenous base is a pyrimidine, and those would be cytosine, thymine, and uracil. After you get your nitrogenous bases, you also need a pentose, which is a 5-carbon sugar. The most common ones you're going to see are ribose and deoxyribose. And once you have your nitrogenous base in your pentose group, you need a phosphate group. And when you put all three of these together, you get your nucleotides. So let's look at some common nucleic acids. First, we have ribonucleic acid, or RNA, which consists of the sugar ribose, so our pentose, our 5-carbon sugar ribose, plus adenine, guanine, cytosine, and uracil. Those would be the nitrogenous bases that you're going to use. And ribonucleic acid, or RNA, is usually single-stranded. The next one would be deoxyribonucleic acid, or DNA, and you notice that NA in both of them is there. It stands for the nucleic acid part. The first letter is telling you which sugar it's going with, which pentose. So deoxyribonucleic acid is going to be with the sugar deoxyribose, plus adenine, guanine, cytosine, and thymine. And you might notice that RNA is going to use uracil as its second pyrimidine, where DNA is going to use thymine as its second pyrimidine. And that's how it always is. RNA is going to use uracil, DNA is going to use thymine. So you need to remember that part. DNA can be single or double-stranded. It is a bit more advanced or complex than the RNA, so a lot of times it is double-stranded, but it can be single-stranded as well, where RNA is usually going to be single-stranded and only rarely double-stranded. Something to remember as well is that adenine always pairs with thymine or uracil, and guanine always pairs with cytosine. And the way I like to remember that is that the curved letters, guanine and cytosine, our abbreviated letters, go together. And then our letter abbreviations that are either made with straight lines, like A and T, go together, or vowels go together. But if you just remember that the curved letters go together, you can always pair up G and C and then pair up whatever's left. And it will be adenine and thymine in DNA, and adenine and uracil in RNA. Genetic information is carried by DNA in all organisms and most viruses. However, some RNA, some viruses use RNA. So, most viruses will use DNA for their genetic material, but some viruses will use RNA. So you can see that nucleic acids are very important for cells. They're made up of a nitrogenous base or series of bases that are going to pair together the same way every time, pentose or sugar, and a phosphate group. And once you get your nucleotide chain and it forms this nucleic acid, you can name it based on which sugar is joined with the rest of it. So ribose formed ribonucleic acid, deoxyribose formed deoxyribonucleic acid, and the nucleic acid's most important function is carrying genetic material. It's going to be carrying the genetic material for all cells and organisms. Plasma membrane. The plasma membrane is also known as the cell membrane or the cytoplasmic membrane. It separates the interior of the cell from the outside environment. So, it being called the cell membrane makes sense because it surrounds the whole cell and keeps it safe from the outside environment. And the cytoplasmic membrane also makes sense because it's holding all the cytoplasm in. It's a membrane surrounding all the cytoplasm in the cell and protecting it from the outside environment. The plasma membrane controls the movement of substances into and out of the cell. So it controls what passes into the cell and what passes out of the cell. So if there are things that are waste and need to get out of the cell, the plasma membrane is controlling that. If the cell needs something, the plasma membrane is going to be what lets it in. And the plasma membrane is composed of a phospholipid bilayer and embedded proteins. Now, a phospholipid bilayer you may not have heard of before. So, let's describe it. The phospholipid bilayer consists of many phospholipid molecules arranged into two oppositely facing layers. So this thing here would be your phospholipid bilayer. So bilayer because it's technically two layers of these phospholipids, and you can see how it kind of looks like it could be making a wall, and this whole thing would go all the way around the cell. Now, these top parts are phosphate groups, and these are fatty acid tails. So you've got phosphate group and fatty acid tails, which are like lipids. So you get your phospholipid bilayer because you have two layers of it. And these phosphate groups facing out bunch together and they repel some things. So they're going to repel what the plasma membrane wants repelled, and then they'll kind of open up and let through what the plasma membrane wants to let through. The plasma membrane also helps anchor the cytoskeleton to provide shape or structure to the cell. So the cell kind of looks like a big blob, or a very small blob, and the cytoskeleton helps give it some shape, and the plasma membrane kind of holds that cytoskeleton in place, because otherwise it doesn't have anything to anchor to hold on to keep that shape. The plasma membrane attaches to extracellular matrix and other cells to help group those cells together to form tissues. So, on the inside, the plasma membrane is holding onto the cytoskeleton and helping give the cell shape. On the outside, the cell membrane is reaching out to the extracellular matrix, to what's outside of that cell, and holding onto other cells to help group them into tissues. Because you usually don't see one cell by itself. Your body is made up of lots and lots of cells, and those cells group together to form tissues, and the tissues group together to form organs, and all together your organs form up your body and make you who you are. So the plasma membrane has a big role in that. Now, something to remember is that plants, bacteria, and fungi also have a cell wall, which goes outside of the plasma membrane. And it blocks out the passage of larger molecules. So the phospholipid bilayer is going to keep out teeny tiny little molecules, but this cell wall is going to keep larger molecules from even getting close to the cell. So you might consider like a piece of fabric the kind of plasma membrane. More stuff can get through it, it's more flexible, where maybe a piece of cardboard would work for the cell wall. It's more rigid, it doesn't bend as easily, not as much is going to get through. Something can get through the cell wall, just as something can get through the plasma membrane. But the purpose is to try to keep out anything that the cell doesn't need or want in it, and to let in what the cell needs. So, plasma membrane has a few main functions you should remember. It is protecting the interior of the cell from the outside of the cell and limiting what comes in and what goes out. It is helping give shape to the cell by anchoring the cytoskeleton, and it is going to join with other cells to form tissues, because cells aren't usually going to be by themselves. So the plasma membrane is a pretty important part of the cell. It's what keeps each cell separate from the other. Proteins. Proteins are large biological molecules consisting of one or more chains of amino acids, which are known as polypeptides. So a polypeptide is an amino acid chain, not just an amino acid, but the whole chain of them connected. Now, proteins have different structures based on how many of them are present. So the primary structure of a protein is its amino acid sequence. So just the one chain, the one polypeptide chain, and what sequence those amino acids are taking. The secondary structure is the regularly repeating local structures stabilized by hydrogen bonds. Now, this is whenever we've got a few proteins interacting together, and the local structures or the regular structures that these proteins will form just all on their own without any help. And some examples would be the alpha helix, beta sheet, and turns. So those are just some different shapes that the proteins can take whenever there are a few of them joining together with hydrogen bonds. Now, the tertiary structure is getting more complex. It's the overall shape of a single protein molecule. So enough proteins have joined together to make a whole molecule, And the tertiary structure is referring to the spatial relationship of secondary structures to one another. So you've got your alpha helices, your beta sheets, and your terns, and it's how they interact together and what kind of shape they all form whenever they form into a single molecule. And you've got proteins joining together, and then more proteins with hydrogen bonds joining together into a larger molecule, and what that molecule looks like spatially. The quaternary structure is going to be the structure formed by several protein molecules. So you can see how we just keep adding proteins here to get different structures. And the more that are added, the more different that these structures are going to look, because they're getting more complex. So several protein molecules are joined together in the quaternary structure, and this is usually called a protein subunit. So it's no longer one molecule, it's multiple molecules. So it's a subunit which functions as a protein complex. So it's going to be more complex, it's going to be able to process more complex functions. And a lot of the time what you're going to see is the quaternary structure of a protein, because you have lots of them together and they are drawn toward each other and then they bond. Now let's look at some of the functions of proteins. We have structural proteins, and these give stiffness and rigidity to biological components that would otherwise be more fluid. They wouldn't have a lot of shape to them. An example is keratin. Keratin is a protein found in our hair, nails, in birds' feathers, and in animals' hooves, and it gives them a hardness that they wouldn't otherwise have. It's not a part of your body like your skin or an organ, and you can feel they're a lot harder than your skin would be. Like your nail and your hair don't feel the same as your skin feels or as your internal organs would feel, and that's because they have this protein present that gives them more rigidity. So structural proteins are going to make things stiffer and give them a harder shape. Next we have enzymes. And enzymes catalyze chemical reactions. So there's a chemical reaction that needs to take place, but it won't be able to take place until this enzyme is present. When the enzyme is present, it catalyzes the chemical reaction and speeds it up and lets it occur. Next, you've got receptors. Proteins that function as receptors bind a signaling molecule to induce a biochemical response. So it receives that signaling molecule and then it binds it somewhere that will induce the biochemical response that needs to happen. Next, you have antibodies, which are also known as immunoglobulins, and these bind antigens and target them for destruction. So an antigen is a foreign body that comes into the cell and it's not welcome, it's seen as the threat, and so these antibodies or immunoglobulins are part of the immune system, and they bind to the antigens or that foreign body and they target them for destruction so that then they are destroyed and removed from the cell and the threat is eliminated. We also have motor proteins, and motor proteins generate the forces responsible for muscle contraction. So think about your motor functions, being able to move your muscles, being able to move around. All of those are able to happen because of these proteins, these motor proteins. So if you're able to move your arm back and forth, if you're able to close and open your hand, those are responsible, those are happening because of muscle contractions, and those muscle contractions are generated by motor proteins. If we didn't have those, we wouldn't be able to contract our muscles like we are. Next, we have pump proteins. So these are proteins that act as a kind of pump, and they transport ions or small molecules across a membrane. So you've got a cell membrane or an intercellular membrane, and the proteins can act as a pump and push these small ions or molecules across the membrane. And that is their primary function as a pump protein, is just to transport these small molecules or ions across the membrane. Lastly, we've got switch proteins, and these act as an on-off switch based on the presence or absence of certain other molecules in the cell. So if a cell was waiting for a certain molecule to be present to do something, then the switch protein is going to act as an off switch and keep the cell from performing that function until the certain molecule it's waiting on is present. Once it's present, it flips the switch and says, basically, it's on. It can perform this function now. So it acts as like an on-off switch for the cell. As you can see, proteins are very important in a cell. They actually can make up a large percentage of the components of a cell. For instance, you could have a cell that was made up of maybe 3% DNA, which is really important. You have to have the DNA encoded correctly for everything else in the cell to function. But the protein could make up almost 50% of that cell's components. So while it isn't maybe as biologically important as that DNA is, it makes up a big part of that cell, and not much would be going on if it didn't have protein there as well, because protein has a lot of functions. So you can see that proteins are very important for cell life and lots of cell functions. RNA, or ribonucleic acid, is made up of a base pair connected by sugar molecules and phosphate molecules. Now the bases in RNA are going to be adenine, uracil, guanine, and cytosine. Similar to DNA, they're going to pair with each other, and each one is only going to pair with one other base. They can't pair with any of the other three. So adenine pairs with uracil, and guanine pairs with cytosine. Guanine and cytosine also pair in DNA. Your curved letters for abbreviation pair together, and adenine and uracil pair together in RNA, and it's your vowels that are pairing together. Now in DNA, you have thymine as your other base, the base that pairs with adenine. But in RNA, you've got a different base. So you can think of it as your two abbreviations that are vowels paired together and your two curved abbreviations paired together. So adenine with uracil and guanine with cytosine. Usually, RNA is going to be single-stranded. But it can form a double helix, like the shape of DNA, by folding in on itself. So you have a single strand made up of sugar molecules and phosphate molecules, and then you've got the little base pairs coming off of it. So it may be like this. You've got your sugar and phosphate molecules, and then you've got your little base pairs coming off of it. And my little lines here are splitting it. So you would have adenine and uracil, guanine and cytosine, adenine and uracil, guanine and cytosine, all the way down. Now, if the RNA was more complex, it may fold in on itself, and this could curve around and kind of form a double helix by being tightly coiled and wrapped together, so that instead of having just one side of the ladder, it curved up and made the one side kind of sandwich it together and form into a double helix that way. But it's not going to be the same kind of pattern, the same structure as DNA is going to be. RNA is also a carrier of genetic material in some viruses. So in viruses, RNA can carry the genetic material, and a lot of times if it's a more complex virus, you'll have RNA that is a double helix. It has formed a more complex structure to carry genetic material for the viruses. In most cells, DNA is what's holding all of the genetic material, but RNA is what kind of moves it around the cell. So let's look at the three main types of RNA. Messenger RNA, or mRNA, carries codes from DNA in the nucleus to ribosomes in the cytoplasm. So it's moving, it's being a messenger and carrying that code from the DNA to ribosomes. Ribosomal RNA, or rRNA, they read that code carried by the messenger RNA. And ribosomes are composed of the ribosomal RNA and proteins. So the ribosomal RNA is a big part of ribosomes. And then transfer RNA, or tRNA, bring amino acids to the ribosomes where they are linked to proteins. So you can see RNA is moving around the cell a lot. It's not just staying in one place. DNA is mainly just going to stay in the nucleus and hold that genetic material. RNA is going to move it around, except in the case of viruses, where RNA is the main holder of genetic material instead of just a transport system. So let me show you what this might look like. So let's say we've got our ribosome, and so you've got RNA, your ribosomal RNA in here. Well, you've got DNA in the nucleus over here, and it's going to get transferred to RNA via the messenger RNA. So it's going to send that code along, and it's going to come into the ribosomal RNA. And then you've got transfer RNA out here, and it's going to bring amino acids into the ribosome. So you've got your ribosome here, and your ribosome holds the RNA. It takes in the mRNA, reads the code from DNA, it takes in the transfer RNA and picks up these amino acids, and out comes a protein. So the whole point of all this was to create a new protein, and RNA is a big part of protein synthesis. So it's going to move the code from the DNA via messenger RNA into a ribosome, and transfer RNA is going to bring amino acids into the ribosome, and the ribosomal RNA is going to take the code and take the amino acid and create a protein and spit it out. So that's what's going on here. Remember the big differences between RNA and DNA? DNA is holding all that information in the nucleus. RNA is moving the information around. It's how DNA gets its code from one place to another. So RNA is more of a transfer system within the cell, and it has several different kinds of RNA so that it can do different jobs in the cell, all working on transferring that genetic code. Viruses. Viruses are tiny, non-living, biological infectious agents. So while they are not technically alive, they are biological agents, and they are infectious. They infect healthy cells. They are going to consist of either a DNA or RNA genome encased in a protein coat known as a capsid. So this capsid is protecting the viral genome until it can get where it needs to go to infect a host cell. And it can only reproduce, this is viruses, they can only reproduce by infecting and taking over a living host cell. So while they are non-living, they must take over a living host cell to be able to reproduce their viral DNA or RNA. Now, once they're inside a host cell, the virus may remain dormant for a while. So it doesn't always immediately enter the living host cell and attack. Sometimes it remains dormant for a while. But Eventually, it is stimulated into the active or lytic phase, and the virus inserts its own genetic material into that of the host. So the host cell is unaware of this, and the DNA that is, or RNA that is mixed in with the host DNA or RNA, it doesn't recognize that right away. So the virus is able to take control of the host's DNA by mixing with it and then taking over it. It causes the host cell to produce many viral genes and proteins. So once it gets in there, it starts saying reproduce, reproduce, make more genes and proteins. And the ones that it are making, the ones that this cell is making are going to be new viral proteins and genes instead of the normal cell's genes and proteins. And then all of these viral genes and proteins combine to form new virions or virus particles that destroy the host cell and are released to infect other cells. So what happens is, once the virus becomes active and it's in the lytic phase, it's going to insert its own genetic material into that of the host cell, so probably into the nucleus. The virus DNA or RNA is going to mix with the host DNA or RNA, take control of it, and then tell it to produce lots more of these viral genes and proteins, which combine to form new virions, and more are being made so fast that the cell can't keep up, and the cell is going to burst or lyse, which is where we get the lytic cycle or lytic phase from, because it is whenever the end result is going to be the cell lysing or bursting open. And once it bursts open, all these virions are released to infect other cells and go and repeat the process. Something to remember is that the genome of both DNA and RNA viruses can be either single or double-stranded. A lot of times a virus is going to not be too complex because the genome is only going to be 3 to 100 genes. It's not going to be very big, but it can be double-stranded if it is a more complex virus. So viruses are sneaky little things. They're going to be protected by their protein coat, the capsid, and insert their genome, their genetic material, into a host cell, take over, reproduce more viral genes and proteins, which form so many new virions that it causes the cell to burst open and send out more viruses into the host to attack other cells.
Amino acids consist of a central carbon atom bound to 4 groups. Now, in every amino acid, 3 of those groups are always going to be the same. They're going to be made up of hydrogen, amine, and a carboxylic acid. So it's the 4th group that is going to differ in the 20 naturally occurring amino acids. And so this 4th group could also be called the designated R. So R basically just stands for any type of chemical chain. And so I'm just going to write here, it differs. Because like I said, although 3 of the groups are going to be the same for every amino acid, the 4th group is going to differ from amino acid to amino acid. So we can classify amino acids into 5 different groups. And so we're going to take a look at those classifications. So the first is nonpolar, which is where the R's are mostly alkyl groups. And so that's like alanine, valine, and leucine. And then we have the polar group, which is where the R's are oxygen and hydrogen and a double bond between carbon and hydrogen, but they are not charged. So that would be something like serine, glutamine, or praline. And then we come to the aromatic amino acids, which is where the R's are rings. And so this is phenylalanine, tyrosine, those are some examples. And then another group is what we would call positively charged, or we could also call this basic. And by basic, I don't mean simple. I mean the opposite of acidic. And so here the R's are amino groups like lysine and arginine. And then last but not least, the fifth group is basically the opposite of this. So it's negatively charged, or we could call it the acidic group. So here the R's are carboxyl groups, and so an example of that is aspartate. So that's a look at the 5 different classifications of amino acids. When carbohydrates burn, they yield carbon and water. And so that's why the name carbohydrates comes from carbon and water. Now, carbohydrates include sugars and starches. And so sugars and starches are the main source of energy that your body uses. So you can see that carbohydrates are very important. Now, there's 3 basic types of carbohydrates I want to go over. First being monosaccharides. It's a really long word, has a complicated spelling with 2 C's in a row. But more important than the spelling is what a monosaccharide is. So it's a simple sugar such as glucose, fructose, and ribose. Now, we call these sugars monomers because these sugars are monomers for more complex ones. And so notice here in monosaccharides we have the prefix mono, which means one. And then I just mentioned that these are monomers, which has that prefix again of mono meaning one. Now you may be familiar with polymers, which has the prefix of poly, which means more than one. So basically these sugars are monomers for more complex molecules. So these are single molecules that are the building blocks to be able to join together with other molecules to form polymers. So remember that because we're going to talk about that more in a little bit. So the second type of carbohydrate is a disaccharide, which is a double sugar such as sucrose, maltose, and lactose. Now sucrose is what we would call table sugar. So it's the type of sugar you use for everyday use, for baking, coffee, that sort of thing. Now the third type of carbohydrate is starches, which are large biological polymers that consist of straight or branched chains of monosaccharides linked together. So let's go ahead and break that down because that's kind of complicated. So these are large biological polymers. So we see that prefix of more than one. And remember, I talked about these being monomers. They're going to join together to form the polymers. And so we see that played out right here because these monomers join together to form polymers. And we even see that confirmed or spoken of again later in this definition. These polymers consist of straight or branched chains of monosaccharides linked together. So starches are basically made up of monosaccharides linked together either in straight or branched chains. Now, in complex sugars, the monosaccharide monomers combine by condensation reactions where water is removed in the form of a hydrogen atom from one monosaccharide and a hydroxyl group from an adjacent monosaccharide. So that's a look at the 3 types of carbohydrates. Some common types of lipids that you are probably familiar with are fats, oils, and waxes. Now, like carbohydrates, lipids are organic molecules. However, lipids have a more complex structure than carbohydrates and are larger molecules. Now, lipids include several different types of groups. And so the first is triglycerides. And so stated somewhat simply, triglycerides consist of a glycerol molecule that is bound to 3 long-chain fatty acids. So glycerol is a polyalcohol and fatty acids are long-chain carboxylic acids. So triglycerides are the most common lipids and make up fats and oils. Now, a second grouping is phospholipids. And these consist of two fatty acids bound to a phosphate head group. And so the fatty acid portions avoid water, meaning they're hydrophobic, while the phosphate head groups seek water, meaning they're hydrophilic. So phospholipids have a structural function. They make up a cell and organelle membranes in your body. A third group is steroids, which are lipids that are derived from cholesterol. Steroids have a four-ring structure with a hydroxyl group at one end and a short carbon chain at the opposite end. Cholesterol is also found in cell membranes and circulates in the blood. Another type of steroids are hormones. Now, the fourth group of lipids that we're going to talk about are waxes, which are formed when a long-chain alcohol binds to a long-chain fatty acid. So again, some common types of lipids are fats, oils, and waxes. And like cholesterol, lipids are organic molecules. A molecule is an electrically neutral combination of two or more atoms of the same or different elements joined by covalent bonds. So I want to go ahead and break down this definition a little bit to make sure we really understand it. Notice up here it says that the molecule is electrically neutral. So that means it has no net charge. So any positive or negative charges in that molecule cancel each other out, so there's no net charge. In other words, it's neutral. Now, it's a combination of two or more atoms. Now, so this could look like this. Two hydrogen atoms or one oxygen atom and one hydrogen atom together would work as well because they have to be of the same or different element. So either one of these works as a molecule. Now, these are some really simple examples of molecules. And so molecules can be simple like those right there, or they can be as complex as large biochemical macromolecules such as proteins, starches, and cellulose. Now, proteins can range in size from 50 to more than 2,000 amino acids linked by peptide bonds, while starches and cellulose molecules can consist of more than 10,000 smaller glucose molecules covalently linked together. Now, there are some substances out there, three in particular that I want to highlight, that you may think are molecules which actually are not classified as molecules. So I want to go over those. So one is aggregations of polar molecules joined temporarily by hydrogen bonds. Another example is electrically charged ions joined by ionic bonds. And then finally, metals consisting of positively charged ions in a sea of delocalized electrons. So these three substances that I just listed, you may be inclined to classify these as molecules, but they actually are not. So I just wanted to warn you about those. So let's just review the definition for molecules again. That's something that's electrically neutral of two or more atoms of the same or different elements, and they're joined together by covalent bonds. So remember all these things. They're neutral. There has to be two, but there can also be more. Same elements or different elements. And they have to be joined by covalent bonds. So generally, when you have a definition of something, it just usually is maybe twofold. Here, there's lots of different criteria something has to meet in order to be a molecule. So you want to be able to remember all these so that you can see a substance, learn a little bit about it, and be able to identify if it is indeed a molecule. Photosynthesis can basically be described as the process by which plants use the energy from the sun to produce food for themselves. But we're going to take a look at a more complex definition of photosynthesis, and we're going to take a look at what the reaction actually looks like that makes that happen. So photosynthesis is chlorophyll containing autotrophs using the energy in sunlight to convert carbon dioxide and water into carbohydrates. So another way of saying autotrophs is by saying primary producer. So this is basically a plant that's not consuming anything to make its energy. It's making its own food and energy. And so primary and secondary consumers are what consume the autotrophs. And so they're not converting sunlight into carbohydrates. They're using sunlight to power, basically, the conversion from carbon dioxide and water into carbohydrates, because light is a form of energy. All right, so this is what the reaction actually looks like. We have light plus 6 moles of carbon dioxide and 12 moles of water to make our carbohydrates. So that's what we're going for, but it also produces oxygen and water, but oxygen is considered a waste product because the goal of this reaction is not to create oxygen, it's to create this carbohydrate. Now, the process of photosynthesis involves 2 stages, the light reaction and the dark reaction. So in the light reaction, photons from light provide the energy to split a water molecule. And the electrons released are boosted into higher energy states and generate reducing equivalents and the energy carrying molecule ATP, which is an abbreviation for adenosine triphosphate. And so we're going to get to that in a minute. And then we have the dark reaction, which we can also describe as the Calvin-Benson cycle. And here, atmospheric carbon dioxide is captured and converted by the reducing equivalents and ATP into first 3 carbon sugars, and then later converted into 6 carbon sugar phosphates, and then into sucrose, starch, and cellulose. So notice here that ATP is used. It's produced in the light reaction, but it's used in the dark reaction. So these 2 reactions are not mutually exclusive. They are dependent on each other. A polymer is a large molecule which is composed of repeating units of smaller molecules that are chemically bound together. So it's a large molecule and it's composed of repeating units of smaller molecules, and we can call these smaller molecules monomers. And so a monomer is basically the subunit or the building block for polymers, because notice here the prefix mono and the prefix poly. Poly means more than one and mono means one. And so this is just one molecule, but when these one molecules join together in repeating units that are chemically bound together, they form a polymer. Now, the repeating unit may be made of one or more different monomers. So some polymers are made up of the same type of monomer, whereas others are made up of different types of monomers. So we have names for those different types of polymers. So the first being a homopolymer, and the second type being a copolymer. So homopolymers consist of only one type of monomer, whereas copolymers consist of more than one type of polymer. Now, one other fact about polymers may form straight or branched chains. So it just depends on the polymer as to whether or not they're forming straight or branched chains. Now, there are 2 types of reactions that are used to make polymers. So one is condensation, and the other is addition. So the way condensation works is it deals with the removal of water in the form of a hydrogen atom from one monomer and a hydroxyl group from an adjacent monomer. So a new bond forms between those monomers. Now, addition works by the electrons of the double bond within a monomer being rearranged to form single bonds with other monomers. So basically, you're altering the chemical makeup of these monomers to form new bonds between monomers. And so that's a look at polymers.